Well, good morning. Welcome to Grand Round. As I was chatting with a speaker, I've invited many speakers over the years, and they're all usually from areas where the weather's more temperate, and they always happen to choose winter day. I think they like to check out how crazy we are, why we live in such an extreme climate, or maybe they just like the fresh air here. So this morning's speaker, Dr. Oliver Fiehn, is from UC Davis. He was originally trained in Japan, got his Ph.D. in Japan, and then worked in the very prestigious Max Planck Institute, where he was the leader of Metabolomics. He was then recruited by UC Davis, and where he is currently professor of the Molecular and Genome Center and the director of Metabolomics. So, as you might expect, that Dr. Fiehn has many accomplishments, and I, of course, have no time to mention. But just as some example, he's on the editorial board of JVC. He's a board member of the Metabolomics Society, as some examples. But most importantly, he is the director, not just a metabolic center, but the West Coast Metabolic Center, Metabolomics Center, which is one of the first 3 such centers funded by NIH for the express purpose, I think, of promoting metabolomics, and in doing so, help us with the research of human diseases in particular and other biomedical researches. So personally, I think that metabolomics is probably one of the most promising areas that has come on board in recent years. Now, many of you know me, but for the few of you who don't, let me tell you that I'm a card-carrying biochemist and have a lifelong interest. My clinical interest is in metabolism. To say, well, that's not surprising. You love metabolomics. But I also would like to tell you that what is really exciting, I think, for us, for this department, is that you will hear a lot of sophisticated approaches that Dr. Fiehn would tell you, but I'm a strong believer that many of these sophisticated techniques will soon be adapted and be used in the clinical laboratories for the routine care of patients, and that is what's really, really exciting. Personally, I got to know Dr. Fiehn because we were funded to do untargeted metabolomics. And of course, we were not equipped to do that, my colleagues and I. So we started looking, and we have a lot of money, we do our due diligence. So what do? We started with the usual suspects. We looked on East Coast, we looked at centers affiliated with the Ivy Leagues, major medical centers, both on East Coast and in the Midwest. But then we found the best is on the West Coast, where Dr. Fiehn is the director of the UC Davis Metabolomics Center. And so we were very happy that when Dr. Fiehn agreed to collaborate with, and shortly thereafter, we were elated to find out that we were right, or he's the best, that he was then chosen by NIH as one of his lab was chosen by NIH as one of the 3 centers of study for metabolomics. So without further ado, let me present to you our this morning's distinguished guest, Dr. Fiehn, who will give us a primer on the fascinating and fast-growing areas of metabolomics. Thank you.
Thank you. Good morning. Well, it was, I was in Minnesota before at the Bollum Symposium earlier this year. At that time, we really had 3 initial centers. Now we have grown to 6 centers, as you can see here. Is this a pointer? No, there's no pointer. Well, is this a pointer? Okay. Well, whatever. So there are 6 centers, including Mayo Clinic, but all of these centers have different focuses. So we are all funded by the so-called NIH Common Funds. And the NIH Common Fund is sponsored by all the different NIH institutes. So there are 50 types of different Common Fund initiatives. And when we were presented, Metabolomics was on the far right side on the lower bottom, so we have to prove our existence, we have to justify what we're doing, and we have to, bring ourselves out to the community, and that's you. So what I want to tell you is whatever I present today, it's available to you. That's the purpose of these 6 centers in green. So including Mayo Clinic, but they, as I said, they have all slightly different angles and purposes. So you can always contact these people and find out what exactly it would be that their specialty, so to say, is. And we have the National Repository for Metabolomic Data at UC San Diego. All the data are transferred to UC San Diego, mandated by Congress. So this time that we can keep the data for ourselves are over, because now we really need to get public. The point how you can access us, how you can access our services, is by 2 means. Either you have money, and you just order a service, and that's what you get. Or you'd say, I want to explore that first. I have a great idea, and I will write a great grant with you, but first I need data. And we call these the pilot and feasibility projects. They're up to $50,000 in services. The call will be coming out in January. Application date is March 1, and we will then review it, general NIH criteria, innovation, and so on. So last year we funded 25% of the applications. So we had actually 12 projects funded across the US. So you can apply to any of the 6 centers, but only to one, with your ideas, and then get funded and get your preliminary data to pursue, great projects in biochemistry, I'd say, and clinical biochemistry. So I have a disclaimer. I got some companies that I'm working with, but I really want to give you some 10 minutes or 5 minutes of background of why we do the things we do. So first of all, you'd say, I'm working in the Genome Center, as Professor Tsai explained. So in the Genome Center, everything is about genotype, right? Genomicists, genomics, new revolution in genomics, everything's so important in genomics. And I am really here on the receiving end, far away, and I have to also justify my existence there. Because the paradigm in the last 20 years was always information flow comes from the genotype to the phenotype. If we would just sequence the hell out of our populations, we'd know about everything there is to know about humans. Well, that's some kind of idea that is out there. Also in my home department, molecular and cellular biology, it's really molecular biology. So similar idea, understand the genes and I'll tell you who you are. There is some truth to it, and I'll come back. That's kind of the theme of my talk. There is some truth, but it's not the only truth. So if you think about there's a lot of levels of integration there, and the point is there's also a lot of feedback regulation. So we, as a biochemist, I would say feedback regulation, but you can also say control, regulation. Yeah. So metabolites would have, of course, a direct impact on protein expressions and activity. This is known, but genomicists tend to forget. And a protein biochemist would always say, well, this is the center of the universe. This is the machine that actually does the job. And so all your genes, you have to have the right folding, you have to be phosphorylated, and if that doesn't happen, all your genes are good for nothing. So there is all this truth, but there's also this environmental component that somehow interacts. And environmental compounds would also interfere with gene expression. Environmental compounds, or external compounds, including drugs, by the way, would also interfere, of course, with protein activity. At the end of the day, if we want to understand disease, metabolite levels, this whole purpose of the cell is to make metabolites. So if you want to understand the disease, it makes sense to look a lot closer to metabolism, because it's basically the output of the machine just before you have a disease. So it's it should be ideally suited for diagnosis. And yes, I mean, for some, you know, in the clinical chemistry, that's what you do. You look at uric acid, cholesterol, glucose, and so on. So you do already have some parameters. Think about metabolic diseases and inborn errors. You know, that's the most often used metabolomic test. Millions of samples across US and the world that tells you about 50 diseases in one snapshot. So some of that is already done quite frequently. And what we try to tell is you can also do it for more hidden diseases that are more complex or chronic diseases to figure it out. So that's the idea why you want to look at, you know, all of metabolites, which we call metabolomics. Or you could say, well, I also want to look at all proteins or all gene expressions. And the funny part is no technology will give you all. There is no single technology that will give you the whole genome. Where there's no technology that will give you the whole proteome. And guess what? There's also unfortunately, no technology that gives you the full metabolome. And not only that, if you think about it, we are really complex. I mean, how can genomicists say that we are the sum of our genes? It's true. I mean, we have lots of SNPs and allelic variations. We have inherited methylations called epigenetics. And yes, there's gender differences and ethnic differences. A lot of that we already know. You know, it's obvious. It's a genotype. You know? But we have a kilogram of gut microbes. And the microbiome is having received a lot of attention in terms of health status. And that is obviously not encoded by the human genome. So we are not even human. We're a jungle. We're a cosmos. So no, I mean, it's really important to think about it like we're not just that. And by the way, a nutritional biologist will tell you it's all about what you eat. Tell me what you eat and I'll tell you what you are, right? So this is really the calorie intake, the food composition, the lifestyle and exercise, and including the disease history. So people who have had certain diseases will be more prone to get another one. So that is all known, but that makes this thing so complicated. If you think about I want to look at the pathways in the blood, I would tell there is no pathway in the blood. What you're looking at is a pathway in some organelle somewhere in a cell, even in an organ of a cell. So there are pathways. If you look now in the peripheral blood, there is a little bit like a footprint of it. You know, and I understand that we want to read everything out of the blood or urine or saliva, something we can easily access. But never forget that we try to do the systems biology or whatever you want to call it. If you do it on the blood, it's really not easy to decipher. But still we have to do. Okay, and then I'll talk about it. So let's step one step back. What do we really know about metabolism? Every one of you has heard metabolism, biochemistry. Some of you have hated it. I'm teaching it. Okay. So the point is, while you get all these pathways here, that's not the whole truth. So this is a figure from this paper in PNAS that we published a couple of years ago, where the editor said we cannot publish this figure because this figure has unknowns. You see, these unknowns have these numbers here, and you can't publish unknowns. And I said, well, this is before and after surgery on ileostomy, so where you basically had changed the microbiome because before there was too much oxygen, and we looked at the ileal effluent before and after in these patients, and that was what we saw in the patients before and after. Afterwards, blue downregulated and red upregulated, and said, well, I see a lot of things, and the mass spec clearly tells me these are all amino acid derivatives. And they must have to do with a change in the microbiome. And of course, we did also we sequenced the microbiome before and after. So we could explain quite a bit, but not everything. And I thought, it's interesting. I mean, these unknowns here are real. They are not just a dream. We can see them, and they might have an impact on the health. So if we then, look at an output of the machine, this is how it really looks like. This is 2 seconds. We have a 20-minute chromatograms, roughly. And in these 2 seconds, you see a lot of peaks, and they are real. They are there. It's not just a data fluke or something. And we can look at them our compounds, and some of them we know, like glutamine. Oh, I know glutamine. I can talk about glutamine for hours. And some people, spend their lifetime on it, right? But I see like there's also others like sugar acids and sugar phosphates. I can't discern it by the mass spec, but I can't tell you the exact structure. I mean, we purchase a lot of like sugar acids and sugar phosphates from Sigma, and that's another one. So there's always more, even in central metabolism. If you then just do the genome reconstruction, these people, would always map it against E. coli and yeast, and says everything is like E. coli and yeast. It's not, okay? I mean, how can anybody say that? So the more they sequence, the less they learn. So we also had another graph where we said like the novel pathway discovered by sequencing. Well, nothing. Okay, so they all look very much alike. And in fact, if you take cells or organs from an organism, they all look different in the mass spectrometer. So it's certainly not true. However, well, classic biochemistry has its role. And Paulson and others in San Diego have spent the last 15 years on so-called flux balance analysis, trying to predict the effect of mutations and onto growth rates, for example, usually in bacteria, but also some in eukaryotes. And what they did is like, if you mutate this gene here and you block it, it will really affect the overall growth rate based on these flux cones. And that is true. This is like glycolysis and pentose phosphate pathway, and if you interfere here, you will have drastic effects. We call those the interstates of metabolism. So there is so much flux on those interstates that yes, you have a giant traffic jam and real problems if there's a problem on a bridge somewhere on an interstate, right? And you know that. Don't know not enough about Minnesota to say which interstate, but hey, now you can get the idea. Now, metabolism has many, many other routes and many, many other streets. Many of them are unexplored, and that's what many of those orphan genes may be doing. Then Mike Snyder and others have published that many small molecules really bind to important kinases. So ergosterol, for example, would directly regulate the activity of protein kinases. And these protein kinases would be signaling kinases. They had nothing to do with ergosterol biosynthesis. So you have to think about evolution, that it would take certain small molecules and use them as a signal, not just feedback regulation. And that is shown here, and we need to think about this more effectively, and that's also, I think, how food may be working in humans, not just as the micro and macronutrients, but also as regulators of our overall metabolism. Now, Joe Noel and others at Salk, I like these Californian dudes. So he's also from California, and he's shown nicely that single point mutations can drastically alter output of metabolism. This is for plants here now, but we're not so far away from plants. I mean, although we don't make everything ourselves, but the idea that an enzyme specificity can be discerned just by blasting and mapping the genome is just horribly wrong. So even if you have a very high similarity, it doesn't mean that your enzyme will do the exact same thing. So you have to be very careful, and it's been shown over and over again that these mutations can have a drastic output differences. Then, while I'm teaching biochemistry, I'm teaching the easy route. I'd say, look at these enzymes, aren't they beautiful? They're all so specific. They go on the interstate and it all flows so fine. Now, this is for undergrads, right? And, well, it's good. I mean, you want to have a clear message for the undergrads. However, unfortunately, that's not true. I mean, many enzymes are just sloppy. Evolution is just as good as needed. It's never going to be perfect unless it's really needed. And in the interstate, because you have so much flux, yeah, you want to have almost perfect transfer of carbons, but not in many other instances. And in evolutionary times, I have to go back to this because it's really fundamental on why metabolism is so complex and why I like it so much. The enzymes are, many enzymes are promiscuous. They carry out more than one function often. And they can use more than one substrate. Both of the substrate specificity and function or specificity can be promiscuous. And you can show this in forced evolution if you do metabolic engineering in the laboratory with bacterial strains. If you want to read more about it, it's a nice review by Tawfik. We have taken this one thing further in an NSF project. We say enzymes are not perfect. Only 50% or less of the enzymes are actually annotated with known functions. And we say many other enzymes will do repair. Everything that can break will break. Many metabolites are high energy metabolites, diphosphates, or they can be easily oxidized and reduced. Guess what? Under stress conditions, they will get reduced when they're not supposed, or oxidized when they're not supposed to do so. Somebody has to sweep up the mess and avoid that these broken metabolites would accumulate and become toxic. So these are repair enzymes, very hard to prove. Oh my God, we do look at lots of knockouts here, and again, there's a very nice review if you want to read about it. But that's what we think a lot of those unknown enzymes are doing. They're not really going new pathways, but rather sweeping up and cleaning up. Okay, so metabolomics now is trying to identify and quantify all metabolites in a given biological situation. However, that doesn't always make sense. You don't always need the big picture or the big fishing expedition, as you get an insult when you write a grant to NIH. Yeah. However, if you have a good hypothesis and you say, I really think it's lipid metabolism, that then you don't need to look at sugars, right? If you say, I'm looking at lipases, I want to knock out these 50 lipase genes, and well, you don't want to look at sugars, maybe, right? So then we call this metabolite profiling, where you more select your machines and your methods towards a certain part of metabolism, and even lipids are pretty complicated. And then, of course, target metabolism. If you really know your biomarker, and you're very sure that this biomarker is actually interesting, you can and should look for it and get your hypothesis proven. And this can be done in very high accuracy because you have stable isotope markers that you can put in, so you're really sure about your quantitative output. And the problem is, if you try to do this for all metabolites, your scope gets much larger, but your accuracy necessarily has to decrease because you can't do it right for all compounds all the time. Then sometimes you have 20,000 populations like gene knockouts or repositories in your freezer. And metabolomics and any of these would use chromatography, which takes time. So if you don't have the time, we do something called metabolic fingerprinting, also sometimes called metabolomics, like quick and dirty, just to find the most interesting cases that are really different by direct infusion mass spec or by NMR or by FTICR or whatever, FTIR. So there are ways to classify samples more quickly, just to get Okay, so now we have all this complex metabolism. We need to measure it, and we do this here with Metabolomics facility. So we have at our center 8 different labs. 2 labs are under my direct control, the Central Service Core and my own research lab. So that has 3,500 square feet with 15 mass spectrometers and now actually 33 staff. And what we've done is we've looked at metabolism and asked what are the best tools that most comprehensively look at parts of metabolism. Okay, so GCMS is one of the things that actually work nice for 50 years now. Very much established. We use the time of flight mass spectrometers that are about 10-fold more sensitive than classic Agilent mass spectrometers. And for accurate masses, for identification of unknowns, we then use the QTOFs. With derivatizations, you get up to 200 identified compounds from specific biological samples. In blood, usually 150 identified compounds. Sugars, hydroxy acids, amino acids, sterols, phosphates, and aromatics. Not all compounds. Bisphosphates wouldn't survive. So NADH wouldn't survive. That's just too hot. A gas chromatogram, has hot conditions. So then if you have those compounds that you say, well, these are specific compounds where we want to have a specific output, you have to go to the realm of LCMS, to the secondary, or we call them specific metabolites, including oxylipids and so on, flavonoids and what have nots, glucuronidated and glycosylated compounds. So they all have to do LCMS. You may have then the need to use special types of methods. If you want to look at lipids, lipids are fine. They all have lipophilic properties. So they really are the best for LCMS, to be honest. We see up to 350 identified and quantified lipids in one run, 20 minutes, actually 15 minutes. Polar neutral lipids from phosphorylcholines to ceramides and sphingomyelins and so on. And we are just discovering right now all these functions for these lipids because people had hard times when they did TLC and scraping it off and put it afterwards in a GC. You can do this, but it's slow. You would never have the ability to screen for thousands of patients in a decent time. So now we can do this since about 5 years or so, or maybe 8 years. And then there's volatiles, and you may not care, but like think about lung diseases, where you would like to look at volatiles. And polymers really are not important, I guess, for medical community. So then if you have all these mass spectra and you want to annotate those, and we have developed databases for those. First of all, here the so-called Fiehn libraries, where we have basically purchased everything we can for our budget from Sigma, put it in the machines, therefore we know the mass spectra and retention indices, and we superimposed it to all biochemistry compounds that we downloaded from CAG databases. And you see there are many more green dots than red dots. The reason is we can't buy all of it. Many compounds are also not commercially available. So we may know about them, but you can't just buy them. If you can't buy them, a mass spectrometrist will always have a problem of identifying it surely and also quantifying it surely. For that reason, we made databases. So here, like the volatile compound database, but in principle, our bin base is also for primary metabolites, where you can see, well, they all share certain mass spectra, the aliphatics and so on, and similar for the aromatics or the, in primary metabolism, the sugars, they look similar by the mass spectra. So we can, even for unknowns, clearly say what type of substructure there are. We have built large libraries, large databases, so to process them always in the same manner, so you get always the same output with database annotations. And then we went one step further and said, since we cannot buy all compounds, and there are so many other compounds that we see in our mass spectrometers, we need to go a step further and predict how they would fragment and how they would look like in the mass spec. And these are the so-called virtual libraries. We have just published in June in Nature the lipid blast library with 200,000 MS spectra validated and so on. I'll talk about that a little further. But now we go on and do other virtual libraries on acyl-CoA intermediates and acyl carnitins and flavonoids. We get this idea further away from just lipids. So why do we look at unknowns? So first of all, we can detect lots of knowns, and if you just look at plasma, heart, and lung, for example, this is a rat study, you'll find certain compounds that would only be found in lung and certain compounds only in the heart. For example, ribulose 5-phosphate, because of the eminence of the pentose phosphate pathway in the heart and not in the lung. So you'll find something that is specific, but also you find a lot of compounds that are ubiquitous between the organs. That's why it's called primary metabolism. Now you also find a lot of unknowns, and we get numbers, and we have the spectra. And since we accumulate them in a database, we can clearly say that these are only found in mammalian systems. They're not found in microbes. They're not found in plants. So because we can say, well, like in sheep, they're always in sheep, these four guys here. But they're never here in citrus or in apples or in soybeans, never appearing there. So we really know that these are specific for mammalian metabolism. So we know, we can approach a little more, and we, in principle, if the experiments are well described, we can at some point also say these are stress-related, or just like heat shock protein at some point was a heat shock protein because some people had it in the heat shock. Yeah. So if you have good metadata associated with your experiments, you will learn also about unknowns, and then you'll get these names. So how do you identify unknowns, and how do we make sure that these unknowns are the most important ones? Here's a study with my friend and colleague Sean Adams on an R1 study where we had 5 different studies, and we combined all 5 studies and said, from all those 5 studies, the meta-analysis of the unknowns that are important in all of those 5 studies, these may be the most important ones to identify firsthand. So we had a type 2 diabetes rat model, pre and post-diabetic conditions, human health, like on weight loss and fitness intervention on postmenopausal women, obese postmenopausal women, non-diabetic. We had muscle mitochondria, isolated them, sparked them with fatty acids to see what happens there. We had then studies on allelic variants in certain African-American women, Gula-speaking African-American women who are very confined in their genotypes. And they had SNPs and UCP3, all the same, like allelic variants and UCP3 and mitochondrial carrier protein. So we combined all those four, and we had like 11 fairly high abundant unknowns that were popping up as interesting in all those studies. So we said, okay, let's give them to our advanced mass specs, purchase for a lot of money, and see what these mass specs can do. So you get these so-called accurate masses. They have decimals. With those decimals, you get elemental compositions. With those elemental compositions, you then go to databases and download what it could be, potentially, with the workflow that we published. I don't want to go into the workflow. We use, for that reason, electronization and so-called soft ionization. For you, those of you who love mass specs, it's important to do because you want to see the intact molecule. If you do that, you will get hypotheses. So one of those was 2-keto-3-deoxygluconate. And this is the molecule with the derivatizations. And we were able to assign a lot of those fragments to the likely fragments that happen in the mass spectrometer. And you would say, hey, it's a very successful annotation. So it took us about $10,000 and 6 months to synthesize this compound. And it turned out it's the wrong one. So this is not true, okay? And this gives you the idea why sequencing in mass spectrometry is not sequencing like in genes. It's just much harder, right? Okay, so it's an isomer. Now I think it's actually the 5-deoxy-6-aldehyde version because we were not able to annotate this little dude here, 144, which is a very strange fragment. But now we say, hey guys, we're wrong. Why don't you try again? Okay. So there's another one where we said, oh, this is easy. This is dipeptides. And, people have ignored dipeptides because they looked at proteins and so on. And I said, look, maybe we purchased dipeptide, it's not the right guy. So you can easily be misled even with the advanced mass spectrometers and the advanced algorithms to predict mass spectra. Not easy to really find the right guys because we have all the positional isomers and so on. Okay, so that gives you the idea why it's difficult. Now, more to success stories. High throughput. And we do high throughput with Stan Hazen at Cleveland Clinic on diabetes and cardiovascular risk, where we, in the NIH Pre-20 project, looked at different human cohorts. And we had to make sure that we can do this in high throughput, really. So at least 2,000 samples, if not more. Also do mouse studies. We have another one. It's the environmental determinants of type 1 diabetes in the young, called TEDDY. Giant study funded by the NIH with 3 countries, UK, Finland, Germany, and US with 4. So where we have 12,000 samples. So again, like I said before, well, you want to finish that in your lifetime. So how do we do this? We really manage these protocols so that they are really, really robust. And that makes a difference between a published paper in analytical chemistry and actually a protocol that works. Okay, you'll find a lot of papers, but hey, that's a papers. You don't take them so serious, do you? Right? So these things actually work. For example, you would have these very abundant ones, and if they get more abundant, because you don't know what the 11,000 sample will look like, right? You want to have some room here at the very high concentration so that it's still linear. So we diluted it a little bit. So we said, no, we need less so that even for the abundant ones, we'll still likely be in the linear range and not just oversaturating the chromatogram and the detectors. We also made a lot of other modifications to make sure that we see all these compound classes, including cholesterol esters that are very, very lipophilic, as well as lysophosphatidylcholines with only one acyl group. They're very, very polar, with all very good peak shapes. You see in the beginning, very polar lipids. At the end, very, very lipophilic lipids. And this is, well, I found it remarkable to be able to do this now in high throughput. These are the internal standards we put in. This is the blood plasma, how it looks like for both positive and negative mode. So that's how we, then get very robust and very high throughput measurements of all those lipids. Now, the identification I promised you, we made these virtual libraries. We made them in a heuristic way. So it's not like first principle, but rather, hey, let's give us 300 different lipids, measure them, and make rules out of it. So we made rules and said, well, you have always this polar head group, always these acyl groups, but you also have other fragments, like water losses. And we can model all those. And so in a spectrum that is called an ion trap, a mass spectrometer, it would look like that. In a QTOF, it would look like that. But in a QTOF, people say, well, there's only this head group and the intact molecule. But in fact, if you look more closely, all the other small fragments are still there. It's just physics. The mass spectrometry community hates it if I say that. I said, like, look, you spent your lifetime, all your different mass specs, and at the end of the day, it's just physics. Okay? So they will be not maybe so abundant, but they are still there. And so don't throw away the low abundant ions. You also then see, and this is also from blood, phosphoinositols with odd chain fatty acids. These do exist. As a typical signaling molecule that's a lot more than you would usually think they are. And this is also the first hit against our library. We validated against 800 publications, so we really took a lot of validation time. We validated our libraries against decoy databases. What if you had proteins or peptides in it? Would they be wrongly assigned as being lipids? We then did a lot of measures of false and true positives and false and true negatives to be sure when we report your lipids to Mike Zai that they are actually most likely true. Yeah, I mean, most likely means like not 100%, but maybe 90%, right? Okay, so then we, of course, put new lipid standards in it, and we validated across 50 different types of mass spectrometers so that it would work independently of the mass spec you do. So it's kind of a standardization in the field. We find the standardization very important. Other types of standardizations, for all our data sets, you will get so-called INCHI keys. INCHIs are just like gene codes, for genes important. INCHIs are the International Chemical Identifiers. It's a small key here that's this long, never longer than that. This is the only and correct way to describe a chemical. The name of a chemical, there are so many synonyms because these people always try to make new claims like I have not glucose, I have D-glucose or whatever, right? So okay, yeah, whatever you name it, there are like over 100 synonyms for dextrose and other blood sugar, they call it, right? So even those, classic ones have a lot of names. But this is the right way. So this is a structural identifier. It comes directly from the structure and it decodes the structure. And then you can compare valine to L-valine because it's very similar. So this is now the official NIH website to do it. You can batch download and batch match across over 200 databases. So you get the link to many, many, many databases, which is like, again, a standardization. So you don't have to worry about like, where do I get now my lipid maps identifier or my human metabolome database identifier, because they're all in there. Okay, so now you have all these different instruments, and now you understand how we can standardize across the instruments. It's by these INCHI keys. These INCHI keys are basically the short version of the so-called INCHI codes. And then you have the structure. Once you have the structure, you can interrogate the structure. You could say, well, compared to my databases, I know certain enzymes, or the databases know some enzymes working on those. And also chemical mapping. If you don't have an enzyme, at least you know how structurally similar those are. If you do it for all the compounds that we measure in our center, this is how our center map would look like. You have lots of lipids, of course, but also lots of other aromatic amines and sugars and vitamins and sterols and oxysterols and neurosteroids and sterol esters and eicosanoids, prostacyclins, and all the funky way can be, from different laboratories, can be now integrated in one map. And we can now show you the differences. If you apply this to biology, that would look how on two platforms integrated would look like. This is stem cells, mouse embryonic stem cells, to progenitor cells. And you directly see that there's a big difference here. Down-regulated are the phosphatidyl and atenolol, especially the not so saturated ones, compared to the very saturated phosphatidylcholines that are upregulated. You see, well, okay, so just by that image, you'd say, well, the desaturase are a lot more active in these stem cells. And also the pathways that lead to cholines. So there are two different pathways that can lead to cholines. One is methylations on the atenolol, and the other one is direct coding pathway. So you can directly, from these images, get some hypotheses. And with those hypotheses, you can go further. And that's what I want to show you now in how we apply this idea to integrate metabolomics with genomics in medically relevant, I hope, instances. Okay, so this is drug treatment. So many people say, I will want to have a therapy for my disease, usually for blood pressure, for example, which would be atenolol or other beta blockers. And this is a standard way of doing medicine, so that's what I do. The problem is many drugs don't work for the patients to which you prescribe them. And that's well known. Not in the general public, but in the medical community, it's well known. The drug, some will respond as you want, and some others will not, or have even adverse reaction. And the point is, we would like to know beforehand who will respond when they show a baseline at the clinic, so that you say, well, you are belonging to this cohort, and you are better served on the other cohort. You have the other genotype or the other metabotype, we call it, or so you would either get a higher dose or a different drug, or So we would like that's the idea. So we want to stratify now our cohorts based on the overall genomics. And so all the studies were done before and after treatment. Followed all the study designs for all the studies I show. This is a study that we did with University of Florida, Julie Johnson, in the realm of the Pharmacometabolomics and the Pharmacogenomics Network of the US, led by Rima Kaduradog from Duke. So this is like the first one where we had baseline assessments, and then we followed them over several weeks, 4 weeks usually. And these are the inclusion criteria: age 17 to 65, untreated home diastolic blood pressure higher than 85 millimeters, and clinic diastolic blood pressure higher than 90, and so on. So no other cardiac disease, and you have always these inclusion-exclusion criteria. And when you then give beta blockers, it's well known that Caucasian people would respond on average, and black people wouldn't. African-American people wouldn't. Well, that certainly cannot be the skin color, right? I mean, that is just the good, easy way to discern genotypes before sequencing. Okay, so before sequencing, you just look at a person, like, you got a black color, you go this way. You got a white, you're a whitey, right? That today I think we can do more sophisticated stuff. So that's why I think maybe we should use genotyping too in clinical decision making. Okay, so if we now apply metabolomics to these cohorts, in the Caucasians we would have different levels in the responders on fatty acid metabolism, whereas in the African American we wouldn't. Okay, so that gives you the idea that something is there different in lipids, in lipid metabolism. What is it? I mean, it could be many things, but one of the major things that we do with lipids is break them down using lipases. So if you then look in lipases, there's even in the protein database a lipase that binds atenolol. And so that gives you already some kind of mechanistic idea why certain people might have better response to atenolol than others, because it would bind differently or differently strong. So they might have a better ability to actually go to the center of the action. So if you do that, then you can understand why we would have a differential response in those lipids. And you get now a hypothesis. From the metabolomic data, you get the hypothesis, why don't you look at lipids and lipases specifically? So if you do that, then we looked in the SNP genotypes. And we found 8 SNPs in this one specific lipase, the phospholipase A2G5. The p-value would never have been sufficient in a general fishing expedition as a GWAS study, because because you look broadly without function, you need 10 to the minus 70 p-values, and even then in the next cohort they will die out. So that's why the GWAS studies always explain only very little bit of your phenotype, because there is no function. So we say function first. Okay, in this case metabolic function, right? But in principle, you could have other functions that you look at. So if you have a phenotype, you derive hypothesis, then you go for the SNPs, you get much higher So this actually is in the literature. There was already implication that this lipase is implicated in the progression of atherosclerosis. And then there's another one, the phospholipase D1. So there are many different lipases, but they all don't have the same function. So these are very specific ones. Okay, next thing we would look into antidepressant. So we did many different drugs actually with 5 clinical centers. And I'll just highlight some of them here. So this is clinical depression. I'm not just being a little sad. So this is really using the categories that the clinical people would use. Selective serotonin reuptake inhibitors. Again, the serotonin that would be taken up in the brain synapses would be made in the brain. So you can't just look at the blood serotonin levels because the blood serotonin levels are made by the gut microbes, and they don't cross the blood-brain barrier, most likely. So you can't just look at the serotonin and then say, well, I'll be able to predict anything here. So we'd say, well, okay, so same design like week 1 to 8 or 0 to 8, before and after, and then looking at the differences in the responders to the non-responders. And there were some amino acids, specifically glycine, that were very highly correlated with a good p-value to the response and non-response. And again, we took that as a hypothesis. So what enzymes might be working on glycine? And that's not so many enzymes, right? So if you then look at the enzymes that work on glycine, there's, for example, glycine dehydrogenase that would clip off glycine and one carboxyl unit, and you would add one methyl group to folates. So it's a direct link to folate metabolism, a direct link to epigenetics. So you have, well, for those people who are really, really interested, there's your other hypothesis. Like maybe that has to do to predicting responses, that something is here different in the epigenetics. Now, for one of those, we found a SNP that was significant at 0.008 value. And this one we replicated in a different cohort. So because now we had function first, hypothesis-driven, looking for the SNPs, then your SNPs will survive in the next round of validation studies. So we started here with 529 responders, and then we did, well, obviously not me myself, but the Dick Winchelbaum at Mayo Clinic. I have to, credit to give the people, right? So at least in the consortium, we are able to validate that. And now we are following up with more mechanistic studies on this depression. So the last study I want to present to you is on statins. And statins are widely prescribed. And actually the American Heart Association says everybody should go on statins. Oh my God, we love it, we love it. I mean, your risk of heart attack would totally decrease, and everybody, and I think the pharmaceutical industry is very happy to hear that. Okay, so there's a problem with statins, of course. So statins really block HMG-CoA reductase, which is on the heart of primary metabolism. So it's really, at the starting or the committing enzyme towards cholesterol biosynthesis. And if you block here, you will have, of course, a lot of feedback regulation to acetyl-CoA, which, as you will remember from your biochemistry lectures, acetyl-CoA is just the big thing in the middle, that connects different pathways. So if you do something here, you will get adverse reactions, and you will get a lot of mess, and you will get a lot of side effects. And this is known. I mean, there are a lot of, side effects being described for statins. I mean, people say, well, still better than a heart attack, which is, I guess it's true. So we still wanted to know for who will these statins work and for whom will they not work, and what are the differences, in the responders to the non-responders. So one of the differences was clearly in the amino acids that are a part of the urea cycle, but that's not about urea recycling or ammonium recycling. It's about nitric oxide signaling. So because arginine, that is the major component here of the urea cycle, is the major building block for nitric oxide signaling. And nitric oxide signaling has been implicated before in the importance of cholesterol biosynthesis and regulating of blood pressure and other overall phenotypes. So that is a clear difference between the responders and the non-responders. But as I promised you, there are a lot more. So first of all, well, there are, of course, the data would show what the clinics also tell. Like cholesterol levels will be lowered. Good for us. So here's the free cholesterol in the responders, lower. Good. Cholesterol esters, lower in the good responders compared to the poor responders. That's nice. Yeah. Yeah, some free fatty acids would be actually increased. Maybe that's not so nice in the poor responders. And then you'd have phosphatidylcholines and ethanolamines that are actually decreased. That's weird because these are not cholesterol. Like that's a kind of like the regulation. I said you inhibit acyl-CoA or do something there, you will see some other effects that is very hard to explain. Okay, so going on, we now want to know what happens at baseline. So these are now the people at baseline when we look at them. And they're at the good responders before the therapy, there's a clear correlation to bile acids in terms of how they will respond to the treatment. Okay, bile acids, as you know, are catabolites of cholesterol. So, okay, that's maybe makes some sense. We also, of course, have some, just want to highlight it, that some metabolomics can give you direct implications from certain enzymes. So statins will also, for example, block this delta-5 desaturase. So, you know, I mean, there's a lot of unintended consequences that metabolomics can show you. But the bile acids were kind of interesting. So we said, okay, so let's look for SNPs in cholesterol and bile acid metabolism and see if we get anything there. And indeed, from all those metabolites as well as other bile acids, we found the glycolic acids to be significantly different. And we then found SNPs for a certain liver transport protein, the solid carrier organic anion transport family member 1B1. So a very specific SNP, again. And this one is implicated in the clearance of bile acids from the liver, and therefore, again, gives you an idea why, you know, these responders might respond compared to the non-responders, because there is already the mechanistic link to bile acids. And indeed, this organic anion transporter had already been shown to also transport statins. Yeah, so there's a lot of things in the literature, but you can't connect the dots. And that's where the science really happens. It's not just the data acquisition, but it's like the data acquisition also, formulating hypotheses, looking at secondary lines of evidence here, genomics, and then going back into literature and see if the literature would support now, you know, your hypothesis. And then you'd say, well, then let's afterwards do a follow-up study in a bigger cohort. And this was done with Ron Krause at the Children's Clinic in Oakland. So if you want to know more about this, I mean, details, these are the papers we've published in the last, 2 years about, so Google Scholar, it's a really good way. So just type in my name in Google Scholar, scholar.google.com, and you'll find those. Much better than PubMed, by the way. Okay. So with that, I'd like to conclude. So I gave you an idea about a lot of technologies in the West Coast Metabolomics Center. It's here for you. We can do services, but we also love to do collaborations. We love to write grants. Oh my God, we love to write grants. And so do you, right? So we've shown you that we, do a lot of new funky methods, and some of them work better. And we'll, in my research lab, we try to make these algorithms better, and that we can actually identify these unknowns, right? So with the genomic data, we think it's hierarchical integration. Like you get a hypothesis from one level of omics, and then you validate it with the other level of omics. So one level after the other, not just giving like, I just throw it all together and see what happens. That's not a good idea. And then with the pharmacometabolomics, it generates hypotheses that you then validate by screening for specific SNPs, and therefore overcome the curse of these high numbers of data that you get from genomic sequencing. With that, I'd like to thank my colleagues, cheminformatics done by Gerd Wohlgemuth and Tobias Kind, the mass spectrometry by Bill Wyckoff, Tobias Kind, John Meissen, Koichiro Kouchi, Tom Chaika, and the Pharmacometabolomics Consortium. I mentioned them along the talk, but it's really Rima Kadura Daug I thank the most, who really relentlessly, pushes the people a lot on the phone. I'd like to thank the NIH for funding it, also European Union to talk about that project. And my disclaimer about Agilent and Alico, I've also worked with them, but also with AB Science. So I'm very happy to answer questions or receive comments. Thank you. Yes, please.
So welcome back. We met you in summer when you were here. So my question to you was, you talked a lot about amino acids and lipid metabolism. How much do you do of the TCA cycle, glycolytic metabolites and stuff?
Yeah, all of them. So some of them are easy and some of them are not easy, right? So like fructose 2,6-bisphosphate is a regulator of the second step of. So this is more difficult. And then if you look in blood, there is no bisphosphates. So because it's a carrier, so it's not, phosphates are not exported. So sometimes I receive this email like, why didn't you find the phosphates in the blood? And I said, well, because it's the blood. That's why. Yeah. So it really depends on what type of cell, what type of organ, and some of them are low abundant. Oxaloacetate, very low abundant. So compared to say malic and citric acid.
So do you focus specifically on cancer metabolism?
Yes, I didn't talk about cancer metabolism, but we have like 4 grants on cancer metabolism. I love cancer metabolism because it's so super hyperactive, right? I mean, compared to chronic diseases, cancer is a beauty to work with. Yes, and we have published maybe 8 papers in cancer metabolism. We also do now flux studies. One of the pilot grants is with University of Chicago, where we look at ovarian cancer cells. Ovarian cancers are really crazy. They eat the lipids instead of the glucose, and we find it pretty fancy. So we look there at using isotope-labeled studies to see why those tumors do that, and not like all other tumors the regular way with glutamine and glucose. Glutamine is very important, for example, in cancer cell metabolism. So we do all that. It's kind of fun. Yeah. Okay.
You emphasized several times the difference between looking at the blood and looking at the cells.
Yes.
And now we have access very frequently to different types of cells. Do you think we'll ever get to the point where we're using these cells that we can now get in a clinical sense to do metabolomics on the cells themselves from a clinical point of view?
Yeah, I mean, it's there are people who I think tomorrow we'll submit a grant on U54 on that question. We'll see if you review us, please. Where we do exactly that high throughput phenotyping of those cells and populations of those cells, and including like, can you do single cells? How sensitive can you be, and so on. So we'll try our best, but cells are not organs, and organs are not complete bodies. So I usually say, you learn things from every level of biology or a study. You learn something, and you have to do a little. You have to go to the other level. You can't just say, oh, I did these cells. No, let's do more cells. So if you've done the cells, you need to go to the next level, and it could be a mouse. It could be a xenograft mouse, for example. I love xenograft mice. But then people say, well, xenograft mice are stupid. They are not people. You need to look at people. Oh, yeah, right. So let's look at people too. I mean, yeah, so then you learn from every level, you learn something, and you just have to bridge the discipline and say, well, I'm a cell biologist, but let's talk to my mouse colleague next door. Yeah? And that is, I think, what we have to embrace. Clinical chemists or clinical clinicians, have to, talk to the basic researchers and say, hey, you're so basic. Well, let's still talk. Yeah. And this is important, and the language is different, and what is important is different. I mean, when I went to the American Heart Association, I couldn't believe they didn't talk about genes at all. It was like 18,000 doctors and me, and it was really freaky. But hey, I mean, that's what I thought. Language. Okay, so we need to learn to speak the other language and to see why people say that this is important and where we need to do this inclusion criteria. We need to do the 3,000 metadata analysis with 5 different studies, and and I say, whoa, wait, we need to look at the individual patient, because there is no general population. Yeah, so there's always these different philosophy concepts, and then we need to talk about it. So, like, what are now the right studies to do? Is this good study the 30,000 Framingham study, or is it a good study with one person following over then the other persons, maybe 10 people following longitudinally, or maybe 100 people, So what is, from which study will we learn more? Well, obviously they're complementary, right? So anyway, there's more questions over there.
I was just wondering what variability you see among some of these samples. For example, if you take a sample from someone's blood sample, morning, noon, and night, you're looking for the same thing, or the profile, does this change?
Oh, yes, of course.
Daily, monthly?
There is this diurnal cycle, but I don't think diurnal cycle is so important. It's more like postprandial and preprandial. So this is often not recorded. When the people show up at the clinic, you tell them to be fasted, but they're not. They didn't think that the donut is food because they do it all the time. So I don't know. Also, people, ask people if they're taking drugs. Do you take any medication? No. Big aspirin is medication, ibuprofen. I mean, so they do have impact. But that people say, no, I take this every day. It's not a medication, is it? So yeah, I mean, there is variability. There's also, of course, technical errors. I mean, if I said it in the beginning, like metabolomics is fantastic, but it doesn't give you the absolute correct value all the time for all compounds. So for some compounds it's better, and some others it's not so good. Yeah, so it's always good to have a good hypothesis. Let's put it this way. Yes.
I was wondering about, just because you're just mentioning when you measure the variability, the role of developing standards, heavily labeled compounds, which, I mean, for this, when you're detecting unbalance, if you'd like to get accurate measurement, do you have part of your program for the development of standards?
Yes, the NIH, actually, not me, not myself. The NIH has funded 2 centers specifically for the metabolic community. And by this way, I have to say, please make recommendations of the compounds you would like to be made. So the NIH recognizes that sigma has failed, and therefore they say, hey, the community should make recommendations of which compounds to make. And so there's one at RTI in North Carolina, and the other one is SRI in Stanford in California, and these 2 centers make chemicals for us. Anyone can nominate. In the nomination procedure, we have to give a reason why. It could be, hey, I need this labeled compound for my quantification, or hey, this is the oxylipid. I've read this article. It's so important in signaling of my pathway. I really need that, and it's absolutely critical for all my colleagues in this field of X, Y, and Z. Right? So you need, and then we have, of course, a committee looking at that. So you better make some good reasons why you want to have it made. Not just like my lab, but rather my lab and 100 other labs, right? But yes, the NIH recognizes this, and that's why we have these chemical standards synthesis centers.
Because talking about other species, have you thought about other species? I mean, a lot of these things can't be measured in blood.
Yeah. Yeah, I have a green background. I'm a plant dude before I came to the US. Actually, I was in Germany, by the way, not in Japan. Max Planck, Germany. But I do now work with Japan on plants and algae metabolism because, like NSF funding, they can't go for mouse and so on. So that's we have also, like, in our services, we serve anyone. So lots of Monsanto and Syngenta and all the other big giants would come to us, and so we have a lot of plants also there. UC Davis has this plant background too. Maybe we are the Aggies, like. Yeah. Okay.
Okay. Well, thank you very much. Thank you, Dr.